Future plans of NASA and ESA include manned long space flight to Mars. But how does the human body actually survive such harsh conditions in space? And how does it actually adapt? And what do we need to consider for a long space flight in terms of safety and long-term risk for the human health? All these questions and much more we are going to cover today, so let's get into it. The movie Fly Me to the Moon with Scarlett Johansson and Channing Tatum just released, and we all get excited when it comes to space and its mysteries. Till today, 559 people flew to space, and we all know the spectacular videos and the images of astronauts being in space and having fun on the ISS. The truth is, space flights have a tremendous impact on the human body. The space environment is very harsh in terms of multiple factors such as confinement, exposure to microgravity, space radiation and noise, as well as isolation. So in today's video, I would like to discuss with you a very famous NASA twin study. You did not hear of it? Yeah, then let's change it and get started. Today, I'm going to present the findings of a NASA study published in the Scientific Journal of Science called the NASA Twin Study, a multidimensional analysis of a year-long spaceflight by Francine E. Garrett-Beckelman and many other people. The NASA study took place between the year 2015 and 2016 and just released in 2019. So you can imagine how many parameters have been measured and how long actually the data analysis and assessment took place. So a lot of time went in and there's a lot to discuss. So what was the objective of the study? We know from previous studies that spaceflight has a detrimental effect on the human body and the human health, spanning from performances to physiological changes to cellular changes to also molecular processes that change. Other reports include previous effects such as bone density loss, effect on cognitive performance, microbial shifts, and alterations in gene expressions. The problem that we have is the data that we have right now is not really good, especially for long-term space flights. Right now, only 8 people out of these 559 people had a long time in space. But the problem with these studies that have been done before is that usually they did not really integrate simultaneous effects on multiple systems, and the data types that were generated maybe not in the same subject. So it's kind of really difficult to assign any real differences. And also the spaceflight duration was sometimes only restricted, or the stay on the ISS only for 6 months. So no one really stays longer than that. So in this study, they report the first integrated longitudinal multidimensional study and spanning of 340 days on board the ISS with a little twist. So the subjects are twins. That's why the NASA Twin Study. The idea behind using twins is you can maybe subtract or see the differences from spaceflight environment compared to what happens on Earth by having a genetically matched subject. In plain text, both have the same gene signature, and any differences found either in the twin that is in space or the other one that is on Earth can be attributed to each of the conditions, either be in space or in Earth. Stay tuned till to the end of the video where I talk about why this might be a very smart move, but not the holy grail in terms of generality. So how was the study actually conducted? We had a pair of male monozygotic twins, Scott Kelly, Mark Kelly, and one of them, so Scott, went actually to space, to the ISS, for 340 days, and Mark actually stayed on Earth. Both were 50 years old at the time of the study, and it's worth mentioning that both of them had different experiences with spaceflight exposure. That is kind of interesting to note, especially at the end of the video where I talk about the limitations of the study. Now, what kind of samples do the investigators actually analyze? So they actually had samples during the time from both of them, so the whole blood, plasma, the urine, stool samples, and they also performed some physiological tests such as cognitive tests and visionary tests. What kind of assays did they do? Why collecting so many samples, different kind of samples? So what is happening when you zoom in really a lot? So what is changing? Is the DNA changing? Is something on the molecular level changing? So that is called epigenetics. Do metabolites in the whole body change? Metabolomics. We know that our DNA is transcribed to a so-called RNA, and this RNA can be very important for some processes in the cell, and we want to know if some of these genes that are transcribed are actually up or down regulated, so therefore we do so-called transcriptomics. Then of course we have a lot of proteins in our cells. Are they affected? Are some down-regulated, some degraded, some upregulated? So you do proteomics. And then of course you go on the physiological state. What is happening with the cardiovascular system? What is happening with the GI tract, so with the microbiome? What is happening in the brain? So physiological and cognitive tests. During this video, I will guide you through each parameter, and I'm going to present the main findings that are important to highlight. In addition, I will share some data and figures from the paper, but I will not go into much detail, otherwise it will be becoming too much complicated. If you're interested in the full version, have a look in the description box, of course, for some additional references. So now at the beginning, we are zooming really deeply into the human body on the cellular level. So I'm talking right now what is happening in the cell. So when Scott flew to space, he experienced, of course, on every level something. So right now we are looking at what is happening on the cellular level. And what they figured out is when they were analyzing his whole blood, where there are also cells inside, right? The immune cells, or the so-called peripheral blood mononuclear cells, which are B cells and T cells, for instance. And they were looking at these. So how do they actually behave during the time? And you have to imagine, if you are feeling cold or if you're having fever, your body is actually really adapting, starting to actually meet the environment. It needs to change in order to survive. So of course, this happens on the very basic level, which is the gene expression. So gene expressions means a stimuli from very outside comes and tells the cell in a very easy way. I'm explaining now the easy way, not the very complicated way, that through some mechanisms it has been told, hey, you need to adapt now to this environment, so please, you need to express a certain gene that has some encodement for a certain protein that you need to actually adapt to the specific environment. And they were analyzing how Scott's environment kind of changed, because space is very harsh, so you can already think of maybe, are there changes, are there not changes compared to Mark? In total, more than 9,000 genes were actually identified that had different expression levels compared to his counterpart Mark on Earth. It was really interesting because it was on one type of cell, or that genes were differently regulated. Some were more in the pre-phase, some others were in the mid-phase, others were in the post-phase. So it was kind of distributed. And then, of course, there were changes in the mitochondrial RNA, which also gives us a hint on maybe the energy production in the cell is kind of impacted. So, and they kind of see an overall large shift. So 6 times of gene regulation in Scott in this whole year. So when he was for 1 year at the ISS, the body needed to adapt to the environment. And that is only possible by changing certain gene expression levels, and that is what they have seen. So the overall finding that they were suggesting is that long duration space flights actually have an extensive impact on the gene regulation in our body. We remain on the cellular level and look more into detail at the DNA level. Now we shift our focus more to the so-called telomeres. So when you have a chromosome in mind, it's actually the little part down there. So what are actually telomeres? So telomeres are repetitive DNA sequences at the end of the chromosomes, and they protect the chromosomes from damage and also prevent inappropriate DNA damage responses. Telomere length is naturally shortened by cell division, aging, and environmental factors such as stress and radiation. So we have a little protein, an enzyme, that has a really cool task. It's called telomerase, that is responsible in extending the telomeres, so by adding a specific DNA sequence to the ends of the chromosome. Kind of nice to know what telomeres are and that we have this telomerase, but why is it important to look at the length of the telomeres? Now, why is it important to look at the telomere length? So kind of nice to know what telomeres are and telomerase doing. Well, the shortening is a hallmark of cellular senescence and organismal aging. Accelerated telomere shortening is actually related to age-related diseases, making therefore the telomere length as a significant biomarker for aging. What is a perfect biomarker for aging? So the American Federation of Aging in 2016 said a good biomarker would be predicting the remaining life expectancy better than the chronological age. 1 point. Second point is covers actually the aging process independently of any disease process. We have small to non-invasive measurements and should be robust for each individual. And the testing should be applicable in animals and also in humans. This sounds in the very first place very promising, but there are caveats to take into consideration when it comes to interpreting telomere length in the context of aging and disease. We have sex and age-related differences. So short telomere lengths are associated with higher cardiovascular mortality risks in females. And when you look in males, age-related telomere length shortening rather than the baseline telomere lengths correlate with an increased cardiovascular mortality risk. In addition, very worth mentioning, of course, is some people and some studies show that telomere lengths do not really have a predictive power of age-related morbidity and mortality, suggesting that telomere length itself may indicate healthy aging, but is not a strong predictor of survival in older adults. Recent studies indicate that telomere length alone provides just a rough estimation of aging and is not really a reliable clinical marker for age-related pathologies. Other biomarkers such as immune parameters and indices of epigenetic age might be better predictors of the health status and chronic disease risks. Another limitation that I would like to give you is more on the technical side. So if you want to measure the actual length, it's done in two ways. The one technique requires a lot of high DNA amount, and usually sometimes the outcomes are overestimating the length. And in the other method that is used, you need really fresh samples and usually is very high labor intense. Now, knowing everything, what telomeres are and what is pro and what is cons, what was actually the finding in Scott? When we were comparing the telomere length of Scott and Mark, in Scott we had a significant increase of 14.5% of telomere length during the time he was actually in space. When he returned actually back to Earth, his telomeres, the length of it, was rapidly shortened within only 48 hours and stabilized actually before he flew to space. the scientists were really interested in why this is happening. So they dig a bit deeper into each data set and were trying to maybe find possible explanations why in SCOT we see this kind of lengthening happening. So first of all, what they see is, again, the gene expression profile for certain proteins that you need for these telomerase. You remember the enzyme that I mentioned before, that were important, but also for the maintenance and this packaging for telomeres. So you have to imagine that it has to be maintained, that it remains its structure, but also that it's packed in the correct order is also important. Then a DNA methylation, so I will come later to it, what this means. It changed a so-called promoter region of a gene that actually is part of this telomerase complex. And the telomerase activity itself was lower than compared to that of his twin brother when they were looking at the activity. So what do I mean with activity? In lab assays, you can actually assess how well a protein is actually doing its tasks. And since that is an enzyme, we actually can read out by fluorescence or any other means how well is this protein working. They were comparing how was the telomerase, so the one that is actually doing these DNA sequences, putting it on the telomere end, how well was it performing in the case of Scott and in the case of Mark. And they saw that the overall activity was downregulated a bit in Scott compared to Mark. But one problem, the samples that they actually took in flight, so during he was in ISS, they actually cannot be taken because the telomerase enzyme activity was not working anymore. And they could not really figure out why. Then they figured out, okay, it was because of the samples that when they re-entered Earth, that the conditions are not perfect. So what's the takeaway for this section of telomere lengthening? Telomeres of Scott got elongated during the spaceflight. This underlying mechanism or the potential consequences even, we don't know yet. In other studies, and this is kind of cool, different subjects have been observed. They were mentioning that also lifestyle factors related to metabolic and nutritional status, but also physical activity and weight loss also have been associated with longer telomeres. So we need future studies and research to understand what is the mechanism behind it and why is this happening and what kind of consequence does this have, for instance, Scott or any other person. I want to make an important note here. I told you that Scott, when he re-entered to Earth, his telomeres actually shortened again to within 48 hours, and within months it was actually on the baseline. There has been also critical shortening and even loss of telomere seen. I told you that this shortening is actually very, very bad because this is a predictor kind of for health issues. So we need to really figure out what does it mean if we have someone out there for a very long time. Does these people who stay much, much longer in space actually have later on a higher risk for any kind of age-related diseases? So we need to assess that if this is true or what could be countermeasures to do it. So that is all ongoing. We remain on the cellular level and we want to shift our focus to something different. I told you before that besides confinement and isolation, people in space also have to deal with constantly exposure to ionizing radiation, in this case galactic cosmic ray. You can already imagine might have an influence also on our DNA. Researchers were interested what is happening to the DNA if you would have these cosmic rays. The researchers were interested in how to see the DNA damages, and they were looking at chromosomal aberrations such as interchromosomal translocation and intrachromosomal inversions. So Intrachromosomal inversions means that you have a DNA breakage in the chromosome, so in one single chromosome, and then this piece is actually just inverted back into the same chromosome, but the information that we have is now kind of flipped around, so it's not lost, but it's kind of not readable anymore in that, or readable, but it doesn't make sense anymore. In the case of translocation, which means is that if you have a breakage on a chromosome and another chromosome, it could be that one end is joined to the other end and vice versa, which means that this information is kind of not making sense for that chromosome and the other way around. Now you know what inversion and translocations are. So what was the finding in Scott? In Scott, the finding was that he had more inversion frequencies rather than translocations, and that is kind of consistent what they already know from other space flights when you're exposed to ionized radiation. They also saw pathways that were upregulated in terms of DNA damage response pathways. So our body is capable of in some cases, or in the very most cases, to actually repair all these kind of breakages that happen during the time. There are really smart pathways that help to actually get this nice and clean. They saw that these kind of changes were persisted over time, and they saw it elevated in Scott. The inversion frequency they also observed post-flight, which means the inversions were still ongoing in his DNA even though he was back to Earth, which indicates that the genome kind of got a bit unstable. Reflection that you have from the ionized radiation-induced DNA damage. We need future research to actually understand what are the consequences, because he stayed only for 1 year. So if you go back to the beginning where I said we would need a 3-year flight only to fly actually to Mars, what happens if you even fly back to Earth? So you would be 6 years around in space, even exposed to all these radiation stuff and so on. So what does this mean for our genes and genome in general? Do we have a lot of DNA breakage? Do we compensate this? Actually, people who come from space from such a long flight are actually very sick. These kind of stuff we need to figure out for future studies. We remain still on the molecular and cellular level. So now we've shift our focus from DNA damage to actually the DNA, and we want to see what is happening on the DNA. And there we have so-called DNA methylation, which is a little chemical modification on a base that is on the DNA, and it's a methyl group. This kind of methyl group decides if it's present or not, if a certain gene, for instance, is expressed or not. What kind of role does actually DNA methylation has? So it's important for genomic imprinting. It's regulating the tissue-specific gene expression and regulation. So you have to imagine we all have in our body, in every cell, the same DNA set. So that's all the time the same. We need another genes or another expression level when we want to have a heart cell. We need another one when we want a liver cell. So there the gene expressions are actually regulated by these DNA methylation patterns. We, us and females, have the X chromosome inactivation. So we have 2 X chromosomes, right? One is active, the other one is not active. And then, of course, we have silencing of oncogenes and proto-oncogenes, and overall chromatin organization. Now you know roughly what DNA methylation means. So we want to know what the investigators found out in Scott, and do we see any differences? So they were comparing the pre-flights, in-flights, and post-flight samples with his brother Mark, and they only saw really subtle changes in the genome of the methylation pattern when they were comparing both of them. So the investigators had the hypothesis that maybe we see local changes in the so-called promoter regions of genes involved in environmental changes and adaptation. So what is a promoter? We know that our cells are capable of actually making a copy from our DNA. These processes, we have a machinery that binds to it, and we have two types of these processes. So one is replication, where we actually replicate the DNA, and then we have the other process, which is the one that is now here looked at, where we have so-called promoter regions that are specific DNA regions before the genes that have to be transcribed, which means the gene that has to be now expressed. So the protein we need now to adapt to the environment, we need to actually express this. This is binding at these promoter regions and therefore promoting it that it needs to be transcribed in this so-called RNA. You know, the RNA later on, mRNA, is later on translated into the protein sequence that we know. When they were analyzing the stuff, they saw, indeed, there is changes in these promoter regions, and therefore helping to upregulate biological processes and pathways that are important to adapt to the environmental changes. Finally, we are actually zooming out a bit. So now we are looking at more the physiological change that happened when Scott went to space. So right now I would like to start with the immune system and the inflammation response. So of course, when you look at the molecular level, you might see some changes there, but it's also important to look, do the astronauts, if they are at space, do they have maybe problems with getting sick? And then we should know if the immune system is affected. What was kind of cool and unique here, despite the stress and the unique conditions that we have in space, the T-cell repertoire, so our T-cell defense mechanism, let's say it like that, which plays a crucial role in the immune system's ability to recognize pathogens, they were actually remaining kind of stable at space. Scott's immune reaction was kind of really overall kind of the same, kind of resilient over the time. That's actually a good sign, because then we know at least that the immune system is not very affected by these harsh environments. We would like to see what is happening with inflammation. So they looked at cytokines. These are little signaling molecules that regulate the inflammation and the immune response. And of course, they saw fluctuations of inflammatory related cytokines, but that was more like re-entering back to Earth and post-flight. During the in-flight and during pre-flight and in-flight, we did not see such a response. It was more that when he was coming, so Scott coming back on Earth, and that persisted a bit over time. So that the immune system itself remained kind of stable, and in the inflammation response was very sensitive for re-entering back to Earth. Of course, again, you could look at the genes and how they're up and down regulated, especially genes that are responsible for immune responses and inflammations. And importantly, some of them had a lasting impact. So when they were assessing how he was doing on Earth when he came back, Scott, that some of the impacts still remained for longer of time. So the inflammation response was still a bit ongoing. And what I really find interesting, and that's kind of novel, what happens when you vaccinate someone in space? Yeah, I was also thinking that, like, I would never come to that conclusion. I would give the vaccination, of course, before he flies. But sometimes, and this is kind of good to know, sometimes it's not possible, because you cannot go back to Earth to get the vaccination and then fly back, right? So it could be that they have to vaccinate themselves during the time they may be going to Mars or coming back. They did the very first test of vaccination in flight, and they tested an influenza vaccination, and his primary immune function, so like chemotaxis, antigen distribution and trafficking, they were actually maintained. So there was not a lot of stuff happening that would be different. So what's the overall takeaway message here? The immune system itself is kind of very stable, very resilient in this space environment. And of course, we have some inflammatory responses from the body, especially when they come back to Earth. Now we shift our focus a bit to another physiological change, which has been observed in the gut microbiome. We all know that astronauts have to eat a very different food. It's different from the consistency, from the nutritional status. It's important to notice what is happening on the gastrointestinal changes, especially on the gut microbiome. They saw in Scott that the composition, so what kind of bacteria, fungi, viruses you find, are altered when they were comparing that to Mark. This unique environment that we have at the ISS, microgravity, altered diet and everything, they have a profound impact on the microbial community. So first of all, we see these changes. But the good news is that when he re-entered to Earth again, it actually got back within months to the pre-flight state of his gut microbiome. The human microbiome is kind of resilient and capable of re-stabilizing after being exposed to really harsh conditions in space. Now we add another dimension on the physiological changes that we observe, the cardiovascular system. So what did they see in Scott specifically? They saw changes in the dimension of the carotid artery, which they see during or after the mission, specifically the thickening of the artery walls, which can impact the blood flow and the overall cardiovascular health. Also, long duration spaceflight have an impact on the blood pressure regulation and the overall vascular health. And these changes are crucial since they affect the heart's efficiency and the body's ability to circulate blood effectively through the whole body in this microgravity environment. Another thing that I would like to mention is they observed when they were measuring the blood plasma levels of the ratio between ApoB and ApoA1. So these are little proteins that are important for cardiovascular health, and their ratio is actually kind of important to measure. I will come to that later. So what they saw, this ratio was increased notably in the last 6 months of the in-flight. So in the second half of his stay at the ISS, he had this increase in this ratio when this was compared to more the pre-flight and the earlier flight samples. When they compare it to the post-flight samples, so he was back on Earth, then the levels decreased. These changes they did not see in Mark. So that was specifically for Scott and for the conditions he observed or he had experienced in the space. Why is it important to measure ApoB and ApoA1 ratio? This ratio is in relation with cardiometabolic diseases. As a new Mendelian randomized analysis shows, it provides evidences supporting the significant causal relationship between ApoB and ApoA1 ratio in these cardiometabolic diseases, and therefore serves as a risk factor. And that's why that ratio could actually indicate in identifying the risk of developing such a cardiometabolic disease in participants. Some of these changes that we see on the cardiovascular level, such as the carotid artery changes, so the thickness of it, they persisted even months after returning to Earth. So therefore, the cardiovascular system has to really work hard to actually get back to baseline. So that is also something that we have to take into consideration when we are planning new long space flights. Now we add another dimension, which is the vision alteration. So Scott experiences changes in his vision during the mission. These kind of conditions are actually already known at the NASA, which is called the Spaceflight Associated Neuro-Ocular Syndrome. Usually that one has symptoms like optic disc edema or globe flattening or even changes in the retina. And they were seeing changes in the structure of the eyes from Scott, including retinal nerve fiber layer thickness and choroidal folds. What I kind of find striking is that this neuro-ocular changes persisted months even after returning to Earth. So he still had problems with his vision even though he was back to Earth. Therefore, long-term effects on the ocular effect. Besides the visionary alteration, we want to see another physiological change, which is the cognitive performance. And there they made a test on cognitive performance with Scott and saw a decline in certain areas such as speed and accuracy after the mission. These changes were highlighting that cognitive challenges are associated with long-term space flights. And the space environment, as I said, with microgravity, with isolation and radiation exposure, they have really a profound impact, it seems to be, on the cognitive performance. And therefore, the study actually underscores that we need to further evaluate what is happening on that level and how do we have effective countermeasures to maintain the cognitive function of the astronaut, so especially in these extended missions. Now we already talked about some of the physiological changes, such as the cardiovascular system is changing, the vision is changing, but also the cognitive performance. Now we want to know what is happening actually on the whole body. So what's the body mass, the nutrition, and the diet? So when they were looking at Scott, they found out that he lost 7% of his body mass. This is kind of expected. We already see that in other astronauts when they come back. But they were pretty happy when they see that overall, when you compare it, that he could actually maintain his body mass to a very stable ground, even though he lost 7% of his body mass. And that indicates that what they were doing was very effective in nutrition and exercise protocol in space. And they were also closely monitoring the caloric intake and the dietary compositions, so that they actually receive and maintain the necessary nutrients that Scott, for instance, needs. When they were assessing the differences in his absorption, they saw that certain nutrients were differently absorbed by his gut. So that might be due to the fact that he is exposed to a microgravity environment, therefore actually having an impact on the GI tract and the system. Future studies have to really ensure we have an adequate absorption of essential vitamins and minerals that are important for maintaining the health. They already did a really great job, but they saw some deficits in him. So that is something they really need to work on. They highlighted the need of dietary adaptations, especially because it's such a unique environment, and of course because it's long space duration flights. When they only stay up to 6 months, you might not need to have these adaptations as compared to 1 or 2 or 3 years. Therefore, they maybe have to modify specifically for these long-term space flights the food formulation, so the nutrient availability and absorption, especially in a microgravity environment, is enhanced. They need to also supplement a bit more. Bone density loss is something critical, especially in astronauts. They have to maybe meet more or work on formulations where calcium and vitamin D are better absorbed by the body to prevent bone density loss and therefore maintain the overall health of the bones through the diet that they have in the microgravity environment. Drinking is very important, so drink a lot of water so you stay hydrated. But the problem that we have in space is when you're in this microgravity environment, all the body fluid tends to shift actually upwards, so to the upper body and to the head. So therefore the hydration status is a bit changed, and the fluid balance, because most of the things are more like here, not so in the lower part. And that of course can also have again an effect on the physiological system, like cardiovascular health, as we discussed already before, but also the renal function, so the kidney function is also affected by this. You want to prevent that someone is dehydrating or overhydrating. And of course this water needs to have maybe balanced electrolytes, so that's kind of crucial for preventing muscle cramps or cardiovascular problems. And they actually want to work on this and make better formulations also for that. Now we have discussed nearly all of the very important information that you get out of this study from very multiple angles, either be on the very cellular and molecular level up to physiological changes such as the cardiovascular system, but also changes on the vision, on the cognitive performance, and on the molecular level, of course, on the DNA, on the telomeres, and all other stuffs. It's a study, so we need to talk about the limitation of the study. We have two individuals, which are male and monozygotic twins, so they both have the same DNA. I told you in the very beginning that this is really cool, but the problem is that and in science we want to have everything under controlled mechanisms or controlled conditions. So in that case, the controlled condition was we have the same DNA, so no changes observed on this DNA background. That's kind of good, and we could see like changes that were happening to Scott were not seen in Mark. But the problem is that we cannot generalize it. Then of course we need more genetic diversity, so different ethnic groups, the females we have to male, of course, young and old. Another limitation of the study is only Scott went to space and Mark remained on Earth. But if you compare harsh environment in space and let's say normal condition on Earth, these are not really comparable. So they will be in itself already very different. The ideal case would be having Mark in a kind of like same scenario but on Earth, and I'm not sure if this can be done in some point. I have no idea. Perfect control would be that Mark had to go through the same by simulating it on Earth. What I find would have been found interesting is the study was conducted 2015 and 16, but the overall paper came out in 2019, so you already have kind of this delay of some years in between. So it would be good to have like a follow-up study where they look at both twins and look at like what is the change happening when they remain for, I don't know, 10 years on Earth. Were there massive changes? Did everything come back to normal, especially in Scott, even though he had some changes still ongoing, even though he was on Earth? They were very frank about it in the study that this is more like a hypothesis generating study. So what it means is, don't take the study now and be like, that is happening to us, everyone in space. No, this is more like testing out what happens if someone is longer in space than usually, and that should give other researchers a platform to ask questions that they want to answer. For instance, what are the differences? Why is the human body behaving different in the absorption? So are there changes in the gut microbiome? If yes, what kind of bacteria do we need to maybe emphasize that they actually take over and help that nutrients are better absorbed? It's a general framework where future studies actually can rely on and cite on and see what can be done differently. Something that was completely out and maybe only partially addressed in the cognitive performance was the psychological stress. Usually at the ISS, there are more people there from different countries, but still it feels like you're isolated, you're in a confinement. What kind of psychological stress are you having, and how does it change your behavior in the sense of maybe even developing depression? We need to figure out if the people, even though they are together, feel lonely. The problem is that we cannot do these kind of psychological assessments in space very easily. Now I come to, in my opinion, the biggest limitation of the study is there were no females included. Of course, you cannot take anyone, female twins from somewhere. You need to be ready to go to space, so they have really a big program, a long program of many years, how to practice yourself to go to space. And it's a huge deal for many people who want to go to space, that they have to do these camps and these programs to attend and go through. But I think the overall changes that we see are more attributed to now more the male body. Maybe the findings can be not simulated in females. So it might be, since we females have hormonal changes, we have the menstrual cycle, so we need to know how our reproductive system, for instance, is behaving. In the case of the males, it's not really known what is happening with the testes, right? So how does the sperm is affected? So very boldly said, do people who go to space are actually infertile afterwards? We need to assess that. We need to figure out what is happening. Are you sacrificing a lot of your health in the end if you do these kind of long space flights? Another thing that I think is kind of interesting to notice is women are much more prone for sarcopenia. So how does the body changes? How does the nutritional composition changes? So it might be different for the males, and females need maybe another formulation of the nutrients that they get in these specific food. Maybe they need other supplements on top. Also, the bone density is very crucial for women. So what kind of exercises do they need to do? Is maybe good if they would do beforehand much more strength training. These kind of questions have to be asked and answered. Another thing that I did not find in the study, which I would have loved to see, they were talking about the epigenetic landscape. So the environment having an impact on the genes, right? And I told you that DNA methylation pattern is one of them. And that's only the half story. The other story is that when you want to look at the epigenetic marks, it is also important to look at certain proteins. So we come back to the vision of the cell, so that you have the cell in front of you, and you know that we have the nuclei where the DNA is inside. So the DNA is not really swimming around like that. It's actually packed, and this packaging is done by really small proteins, and they are called histones. So it's actually wrapped around them. And these histones can indeed be modified. And these modifications are actually very crucial, because they are also important, like the DNA methylation, if this region is actually open or is it closed. If it's closed, you can actually not really see the gene that you want to have. If it's open, you can actually see where's the sequence, how can I transcribe it so I get the gene that I want to have, so the protein out of it. That would have been amazing to see if histone changes were observed. So if certain modifications were up or down regulated, because we know now that these modifications are really highly tied to the environment of the cell, of the metabolic state of the cell, and upon changes, the cell decides on, I need to have certain genes expressed or not. And this is kind of important, and I would love to see if they might include it in future in some other studies, because that was kind of one part that was missing on the epigenetic side. We learned a lot today. So we learned that Scott went to space, Mark stayed on Earth, and Scott went through a lot. Space is a very harsh environment with microgravity, with ionized radiation, with a lot of confinement and exposure to many different things. And Scott's body was adapting to the space environment. We learned now how. We learned from the molecular level that it was on the telomere they were lengthening. Then we were looking at the cardiovascular system. How's it changing? We were looking at the vision. We were looking at the cognitive performance. We were also looking at the DNA. So many aspects. And I chose this really cool study to show you how important it is. You have one big question, that is, how does the human body behave in space? It's not enough to answer this question only maybe based on the DNA. Genes are expressed up and down regulated. No, you have to see, okay, metabolites are changing, how is the body behaving, so from the physiological changes. So it's all intertwined, and they really did a great job with many authors, many labs involved that actually do a really great job in explaining what do they see and why these changes might happen. I hope you had a lot of fun that I explained it to you, and I hope that this was kind of interesting. Now, if you got interested and you want actually more, then please check out my other video where I'm explaining 3 clinical trials that will revolutionize medicine this year. And if you want to see more like a podcast episode, then you can watch this episode with a medical doctor. I talked with her and why she actually went into more of the longevity and lifestyle medicine. So as always, let a little dose of honest science in your life and brighten up your day.